So, ladies and gentlemen, you are all welcome to my YouTube channel. So, in today's video, we are going to look at the uh, concept of chemistry of macromolecules uh, for distance learning students. And this is our first lecture series. And my name is Mukhtar Ali from Department of Biochemistry, Ahmadu Bello University, Zaria. So in this lecture, we are going to start with the introduction of biochemistry. So first, we need to understand the concept of biology. What is biology? Biology is the study of life. As we all know, is one of the main branch of science that concern with the study of living things or organism. Bio means life, and biology means study. So the entire biology is trying to talk about the living organisms, whether it is plant, animal, microorganisms, any living organisms is under biology. And of course, as we said, bio means life, and biology means study. So what is life? Actually, to define life is a very complicated debate. And in fact, there is no any single definition of life and death. Rather, the processes are used to describe life and death. So now, what does it mean to be alive? Life can only be defined by a list of properties. So there is no single definition of life, and there is no single definition of death. So we can only find life and death based on the list of properties or some characteristics. So what are those properties that can actually be used to identify or describe life? So the properties of life include first organization, how the living organisms organize. So generally, the organization of life is actually started at the level of cells. But what actually produces that cell? So first, under this organization, we have unicellular. So when we say the unicellular, it means organisms that have only one cell. There are organisms that have only one cell. And of course, we know that cell is the structural and the functional unit of life. Without a cell, there is no life. And of course, if there is any problem with any particular living organisms, it means that the problems is emanating from the cell. So even disease, life processes are all taking place in a cell. So if there is a problem or alteration or any distortion in a cell, it means that there will be a problem. So the life and deeds of each and every living organisms depend on cell. But now, how do we get this cell? So generally, the life itself is started from atoms. So you see atoms, the life started from atoms. And then of course, from atoms, we have these atoms joined together to produce what you call a molecules. And then the molecules also form a monomers. An example of monomers, we have like fatty acid and glycerol. We have, apart from fatty acid and glycerol, we also have amino acids. We have simple sugars. We have nucleotides. So atoms, like for example, hydrogen, oxygen, carbon, and sulfur, they are the atoms that join together to produce a simple molecules. An example of these simple molecules, it can be actually CO2, it can be CO, it can be NH2, it can be, of course, SH2, it can be COOH. So all these are the examples of the molecules that can be produced from the atoms. And of course, these molecules, when they join together, they produce what we call of course, monomers. An example of the monomers of life, we have amino acids. Amino acid, we have amino. We have amino acid. Amino acid are the examples of the monomers. We have nucleotides. We have fatty acid and glycerol. We have simple sugars like glucose. So these monomers join together to produce macromolecules. And examples of these macromolecules, we have proteins. Examples of these macromolecules, we have proteins. We have nucleotides. So the examples of these macromolecules, as I said, we have proteins. Proteins are examples of macromolecules, which is number one. And then number two, another examples of these macromolecules, we have carbohydrates. We have carbohydrates. And then apart from carbohydrates, we also have nucleic acids. We have nucleic acid. And examples of this nucleic acid, we have DNA and RNA are the examples of this nucleic acid. So DNA are the examples of nucleic acid. So these macromolecules join together to produce supramolecular complexes. And examples of these supramolecular complexes comprise virus is part of or is an example of supramolecular complexes. And how do we get these supramolecular complexes? Supramolecular complexes are formed as a result of non-covalent interaction between these macromolecules. So as these macromolecules are joined together through non-covalent interaction. An example of non-covalent interaction includes hydrogen bonding. Van der Waals force of attraction are examples of non-covalent interaction. So when these macromolecules, protein, nucleic acids, carbohydrate, when they join using this non-covalent interaction, that is when the supramolecular complexes will be generated. The supramolecular complexes will be generated. And of course, the supramolecular complexes also combined together to produce organelles of the cell. And from the organelles of the cells, then when you have the entire organelles of the cells, then it will now give you a cells. So ladies and gentlemen, under this organization of life, we should understand that we need to look at it under unicellular and multicellular organisms. So unicellular organisms are actually organisms that have only one cell. And of course, these cells are formed from atoms. And of course, the life itself is started from atoms. So atoms form a molecules form monomers, and the monomers form macromolecules. And then the examples of these macromolecules, as I said, we have proteins, carbohydrates, nucleic acid, and lipids. And then we have supramolecular complexes. Examples of supramolecular complexes, we have virus, because that is why virus, it is a very complicated organisms, and that is why it is not even, or is a very complicated supramolecular complexes, and it is not up to the level of cells, and that is why virus is not considered as a complete living organism, because it is under supramolecular complexes. And another examples of supramolecular complexes, we have prions. Prions are also examples of supramolecular complexes. Prions, prions, we have prions, prions are examples of supramolecular complexes. And of course, even the ribosomes of the cell, the ribosomes of the cells is also another examples of supramolecular complexes, the cytoskeletons. And then, so this is actually how the organization of life starts. And then under multicellular organisms, still under organization, we have cells, and then the cells form tissues, and then the tissues form organs, and then from the organs, we have a complete system. When you have a complete system, it means that you have a complete functional living organisms. So that is under organization of life. Then the next thing, apart from this organization of life, we need to also look at some different types of tissues that are found in a cell. So, for example, now we have different types of tissues. We have connective tissues, we have epithelial tissues, and of course, we also have muscle tissues and nervous tissues. So these are the types of tissues that we have. And of course, under examples of unicellular organisms, as we mentioned under the organization of life, we have bacterium. So bacterium means singular, one bacteria. It means that it has only one cell. And then under multicellular, like in humans, these are the kind of the tissues or the types of tissues that are found in humans: connective tissues, epithelial tissues, muscle tissues, and the nervous tissues are the examples of tissues that are found in man or in humans. So that is the first actually organization of life, or sorry, the first properties of life. And apart from these first properties of life, we also have metabolism. So metabolism is also another properties of life. So what is metabolism? Metabolism is a sum total of all biochemical, or the sum total of all the reactions that are taking place in the body. And there are trillions of cells there are trillions of reactions that are taking place in our body at a specific period of time, or at a particular given time. So billions and millions of reactions are taking place in the body in order to maintain a proper life activities. So life depends on the enormous number of interlocking chemical reactions that are taking place in the body. And of course, the summation or the sum of all these chemical reactions that are taking place in the body is called metabolism. So when you add them, when you sum all the reactions that are taking place in a living organisms, that is what described as metabolism. And these chemical reactions are interlocking, interconnected, and interrelated with one another. And of course, with these millions of reactions that are taking place in the body, if there is any problem with any of these reactions, it means there will be a disease, there will be a problem. And of course, there are at least 1,300 reactions that are taking place in the human body. And this is because at least 1,300 enzymes exist in the human genome, and each enzyme catalyzes at least one type of reaction. And of course, under this metabolism, we should know that uh, the human genome, that is the human DNA, it contains 1,300 enzymes. And each of these enzymes is responsible for catalyzing one reaction. So it means that from these, there are 1,300 reactions that are taking place in the body. So, and of course, this metabolism is divided into two. We have catabolism and we have anabolism. So when we said catabolism, it means it's a breaking down of large molecules of food into smaller ones. So when you break a large molecules like carbohydrates, proteins, and lipids, they actually produce smaller molecules. And as these smaller molecules are produced, energy is also released. So that is why in most of the catabolic reactions, that is breaking down of large molecules into smaller ones, energy are generated in the form of ATP. And then of course, at the same time in anabolism is when you have a smaller molecules joined together to produce a larger one. So in the process of the anabolism, the large molecules are synthesized, and also energy is needed to join these smaller molecules. So that is where anabolism is the energy consuming system, or is the energy consuming pathway. So it consumes energy, while catabolism, it releases energy. So it means that during catabolic reaction in the body, energy are produced like glycolysis and so many others. Then in the case of anabolism, then smaller molecules join together produce larger one, and energy are consumed. So apart from metabolism, another properties of life is homeostasis. And when we said homeostasis, it means the maintenance of the internal environment. There are so many physiological parameters that we have in our body, and they must be maintained. They must be maintained within a stable. They must be maintained under stable conditions. Like for example, if our blood glucose is supposed to be 5 milligram per deciliter, so it should be that. If it's actually lower than that, or if it is above that, then there will be a problem. So that is why homeostasis, like for example, our body temperature is actually supposed to be around 37 degrees centigrade, and whenever there is a problem, so under this homeostasis, the body temperature is supposed to be 37 degrees centigrade. So it must be maintained, and that is homeostasis. That is to maintain internal environment, to maintain the to maintain internal environment. And of course, there is like for example, the blood pressure. It should also be within a constant range. If it is below that or above that, there is must be a problem. So homeostasis is one of the properties of life that each and every living organisms must maintain a constant internal environment, including body temperature, blood glucose level, blood pressure, and so many other physiological parameters. And then the next actually properties of life is growth. So each and every living organism must grow either internally or externally. And when we said growth, it means is the ability of living organisms to actually grow. And what is growth? That is the irreversible increase in size, which may be due to enlargement of a cell or increase in number of cells. Like for example, we as a human, when we are born, the number of cells that we have and when we get adult are not the same. So we grow due to increase in a number of cells for some organs, while some organs is due to the enlargement of a cell. So each and every living organism must grow. And this is one of the actually properties of life. And then the next one, which is number 5, is reproduction. So each and every living organism must reproduce, and then also response and evolution. So when you said response, it's like stimuli. Of course, all living organisms respond to a stimulus. Whether it is chemical stimulus, whether it is biological stimulus, we respond to it. And then evolution, that is how living organisms change with time. So generally, living organisms change with time. So as time goes on, living organisms change due to environmental changes. And of course, evolution generally take longer time before it takes place. Some of the evolutionary process take millions of years, some take thousands of years. So each and every living organisms have a greater chances of undergoing evolution. So this is also another properties of life. So apart from, so these are actually the properties of life. So we cannot define life, rather we can actually describe life based on some certain properties. And here we have 7, and we have so many others. So that is biology. So biology is the study of life. So each and every living organisms possess life based on the following set of properties. And if there is a problem with any of these properties, it means there will be a problem with that particular organisms. So actually biochemistry is mostly rely on the second properties, which is metabolism of macromolecules. So, but in this course, we are actually focusing on the chemistry of macromolecules. So let's actually look at the biochemistry. That is the definition of biochemistry itself. So what is biochemistry? Biochemistry is the study of chemical processes in a living organisms and how they relate with one another. So generally in living organisms or in every living organisms, there are so many chemical processes that are taking place, and these chemical properties or these chemical processes are related with one another. Because in biochemistry, we are trying to study life, or we are trying to study living organisms using a molecules. And these molecules are mainly proteins, carbohydrates, nucleic acids, lipids, hormones, enzymes, and so many others. So in biochemistry, we are trying to study the chemical processes that are taking place in a cell. And of course, biochemistry is one of the very interesting area that each and every student from life sciences and medical sciences must actually understand it and do it at the same time. You must do it and then you must understand it, because that will give you an idea of what exactly taking what exactly happening in a cell, and that will actually help you to manipulate life, and that will actually help you to treat a disease if there is any problem with any particular living organisms, like in the case of human. So each and every disease must actually or have a biochemical basis. You can treat that, you can treat a particular disease from biochemical point of view based on the biomolecules and the chemical processes that are present in that in a specific or in that specific types of cell. So that is that. So of course, this biochemistry, it can also be defined as the application of chemistry in biological processes. And of course, biochemistry is both life science and medical and chemical sciences. Of course, examples of these chemical processes that are taking place in the body can be catabolic and can be anabolic, or both, which is metabolism. So each and every chemical processes that are taking place in the body must either be catabolic, that is the breaking down of a molecules into smaller one, or anabolic, that is combining of smaller molecules into larger one. So each and every chemical processes that are taking place can either be catabolic or anabolic. So you should get that. And then of course we have so many other branches of biochemistry, but specifically for DLC student, we are going to focus more on the area of medical biochemistry. So what is medical biochemistry? Medical biochemistry can be described as a branch of general chemistry. It is scope is not as broad as a general biochemistry, because in general biochemistry we have so many other branches. So in medical biochemistry, we are trying to study the chemical processes that are associated with health and disease. So we are studying the chemical processes or the biological processes that are related with health and diseases. And of course, from here, medical biochemistry is defined as a study of biochemical processes that occur within the human body in relation with their application in the field of medicine. And when we said field of medicine, it means health and disease. So we are trying to study the biological processes in human body and the application of that chemical processes in the field of medicine, that is in the field of health and diseases. So understanding biochemistry or medical biochemistry will actually help you to understand the disease pathogenesis, the disease processes, as well as the health condition of a particular human or of a particular patient. So that is why it is very, very interesting to have understanding of biochemistry. And in fact, if you are a medical doctor or if you are any health practitioner, if you don't have knowledge of biochemistry, it means that you are not actually a medical doctor, because there will be a lot of gaps, there will be a lot of problems. And of course, let's see what are some of the applications of this medical biochemistry. So one of the application of this medical biochemistry can be actually in disease diagnosis. And when we say disease diagnosis, it means identifying biochemical markers for diseases. Like for example, you can have a situation where you have a particular patient and you don't know specifically what types of disease that particular patient is actually developed. So the best way you can actually use a biochemical markers to detect which specific types of disease is that. Like for example, when we said biochemical markers, it can be carbohydrate, it can be proteins, it can be nucleic acid. Like for example, you can actually know whether the liver of a particular individual is having problem or not. Like for example, there are some molecules that are meant only be present in the liver. So now maybe when you actually collect blood sample and you now test those particular molecules in the blood and you now see them in the blood, it means that there is a problem. Although some biomolecules have their normal range in the blood, but when you see that their range goes very high, that indicates that the organ that is responsible for storing that particular biomolecules is damaged, or maybe it undergoes necrosis. It's damaged. So that is why maybe the content of that particular organs is releasing into the blood. So that will tells you that that organs is having problems. Like for example, one of the main biochemical tests that we do in the lab is ALT and AST. When you said ALT, it means alanine aspartate transaminase. It's an enzyme that's supposed to be in the liver. So, but when you collect blood sample and you detect the presence of that enzymes in the blood, it means that the liver it's actually having problem. There are some damage in the liver. That is why it is releasing its content. So that will give you an idea that there is a problem with the liver. Like for example, you can actually test hepatitis with that. But not only hepatitis, because there are so many diseases that can affect liver and that can lead to that content. But it can serve as a first line of diagnosis. That will give you an idea whether the liver is having problem or not. But from there, you can go actually for a specific diagnosis. And at the same time, maybe kidney. If kidney is responsible for excreting a particular biochemical markers, and now we now found out that that biochemical markers in the blood goes very high, and that tells you that it goes high because the kidney is not actually excreting that. So that is one of the application of biochemistry. And then second, therapeutics. So with the knowledge of biochemistry, you can be able to design drugs, you can be able to actually target a particular specific biochemical pathways. Like for example, you can look at it from parasitology point of view. As of recently, we conducted a particular research where we found out how Plasmodium falciparum embedded into the red blood cells. So we were able to identify a particular proteins that is responsible for actually invasion of the Plasmodium falciparum into red blood cells. So if we are able to actually get a molecule that can block the activity of that proteins that have the Plasmodium falciparum to get into the red blood cells, it means that you can of course block the Plasmodium falciparum pathogenesis, and that will actually lead to the elimination of the parasite, because if the parasite cannot get into the blood, it means that they cannot survive. So as a result of that, with this, you can actually use the knowledge of biochemistry in drug development. So with the knowledge of biochemistry, you can identify a specific target proteins. You can also identify a particular compound that can block the activity of the proteins. And that is also a second application of medical biochemistry. Second, personalized medicine. So in personalized medicine is tailoring treatment based on individual biochemical profiles. So with the knowledge of biochemistry, can get the biochemical profiles of a particular individual, and from there you can design and prescribe drugs specific for that particular individuals, and that is personalized medicine. So it's a treatment of one specific individual. So that is a personalized medicine, and it is based on the biochemical profiles of that particular individual. So ladies and gentlemen, I will actually advise you, if this is the first time you are coming to my YouTube channel, please try to subscribe and join the YouTube channel at the same time. And also interestingly, I have an online e-learning that if you want to have compiled videos, compiled write-off, quiz, and some related questions related to this biochemistry, you can register with it, and then you can actually enjoy it. In fact, weekly we'll be giving you some questions to be answering in related to this particular course. In fact, apart from this biochemistry, we have physiology and microbiology there, so you can enjoy it. You can enjoy the websites and have access to all our videos in chemistry of macromolecules, our write-off, related questions available, or related questions that are related to this course will also are also there. So I will advise you, if you wish, you can go and actually register with our website, and I will drop the link for the videos on how to register and get access to our content. So this is this. So the next thing is we should know that there are millions of complex reactions that are ongoing in the human body at any given time. And these reactions can be in terms of the balance of endocrine system. Like, for example, we know that as a human, we have what we call an endocrine system, which is hormones. So when we said hormones, it means that are chemical messengers that tries to break coordination. And these endocrine molecules, they are molecules that are excreted in one particular part of the body, excreted into the blood, and then transported to their target organs. So some of these reactions that are taking place in the body include the production of a particular hormones, the synthesis, the transport, as well as the utilization of these hormones in the target organs. Like, for example, adrenaline is an example of hormones that actually trigger anger. So we develop angers because of adrenaline, and we come back to our normal stage when this adrenaline is actually break down and utilize. So that is why we should understand that there are some proteins in our body that we produce them only when we need them, and we degrade them after they elicit their physiological activities in our body, like adrenaline. And that is why we did not constantly get anger. So we get anger when there is something that trigger the production of adrenaline. And once the stimulus that trigger the adrenaline is being relieved, then the adrenaline will be break down and then it will become normal. So these are the examples of the complex reactions that are taking place. It can also be in terms of the storage and the utilization of fuels molecules such as glucose. Like, for example, if we take excess carbohydrate, we have to actually, it will break down into glucose, and then the glucose will be transported to the liver and stored in the form of glycogen. So there are billions of reactions, millions of reactions that are taking place in the body, ranging from the endocrine system, storage and utilization of fuel molecules like glycolysis, glycogenolysis. All these are the examples of the complex reactions that are taking place in our body. And then in addition to this, so biochemistry deals with the structure and function of cellular components such as proteins, carbohydrates, lipids, and nucleic acid, and these are collectively known as biomolecules. And that is why, specifically for the purpose of this lecture, or for this course, we are mainly going to concentrate on the concept of these biomolecules, known as the chemistry of macromolecules. So biochemistry is also associated with the structure and the function of cellular components. But in this course, we are going to mainly focus on the structure of the biomolecules and the function as well, but mostly on the structure, and that is why it is called chemistry of macromolecules. And these major macromolecules that we are going to look in this course are proteins, carbohydrates, lipid, and nucleic acid. So let's look at how these types of macromolecules are formed. So these macromolecules are actually formed from their monomers. An example of the monomers, we have amino acids. And examples of this amino acid, we have something like glycine, which we are going to look at it later in our lectures. So amino acids generally form proteins, and then fatty acid form lipids, and then we have sugar residues, which can be glycerol, and then monosaccharides. So the sugar residues, which can be glycerol and monosaccharide, can use. So these sugar residues, which can be glycerol, monosaccharide, then form what we call carbohydrates, and then we have nucleobases, which form nucleotide with monosaccharides. Pause. Nucleobases plus monosaccharide will give us nucleotides, and these nucleotides are DNA and RNA. So these are the four major macromolecules that we are going to look at in this course: proteins, lipids, carbohydrates, then DNA and RNA. So then the next thing is, of course, in this course, the main focus of the medical biochemistry is in understanding how these biological molecules give rise to processes that occur within cells, which in turn relates greatly to the study and understanding of the whole organisms, that is, for example, in this case, human being. So you see, actually, from our previous slides, we talk about how the living organisms are formed and how we understand living organisms from the biomolecules. So we should understand that each and every living organisms, as I stated, are actually formed from atoms. And it's these atoms that joins together to produce or to generate macromolecules. Okay, so the next thing that we are going to look at is breakthrough in biochemistry. So the first scientist is actually the Watson and Crick in the year 1953. They are the first scientists that was able to demonstrate the three-dimensional structure of DNA. And these are the two scientists, James Watson and Francis Crick. They are the two scientists that was able to clearly demonstrate the three-dimensional structure of DNA. And of course, the Rosalind Franklin is a scientist that was able to demonstrate the structure of the DNA using X-ray crystallography, followed by Linus Pauling, which was able to actually demonstrate which is actually the great pioneer of molecular biology. And then the last one is, of course, is Elizabeth Blackburn. Is actually the scientist that was able to discover telomere. That is telomere technology. And of course, when we say telomere, is the tail end of the chromosome which protect the integrity of the DNA. And of course, the longer the shortening of the more the shortening of the telomere, then the old we get. And of course, if we are able to maintain the integrity as well as the structure of the telomere, then it's actually slower age and aging processes. So that is why it is very, very important to understand that we get old, we get aged as our telomere shorten. So that is why now scientists are even trying to reduce aging processes by lengthening telomere. And of course, as we continue with this lecture, we are also going to look at the concept of this telomere technology. So then the next thing that we are going to look at here is, of course, the human genome. So what is human genome? The human genome is the entire set of the genetic material of a living organism. So the entire set of the genetic material of a living organisms, which is a DNA, is what you call human genome. And this human genome is, of course, the DNA, because DNA is the genome. So when we take the entire or the complete set of the genetic material of human being is what you call human genome. And this human genome is DNA, which is usually found in the nucleus. And although apart from the nucleus, we have some other organelles that these DNA are found. We have mitochondria. So in human, DNA is present in mitochondria and also in the chloroplast. So this DNA, it carries a complete genetics information responsible for the development and functioning of an organism. So you see, this DNA is very, very important. This DNA is what makes us to appear the way we are. This DNA is what makes us to be intelligent. This DNA is what makes us to be differ from one another. And of course, this DNA, in simple terms, we call it the language of life. You are the way you are. I am the way I am. The other organisms, the other species of organisms are the way they are is because of this DNA. So DNA is like the signature of life. It's what make each and every one of us differ from one another. And of course, while the sequence of bases is identical in all humans, so we have a sequence of bases. These bases that are found in humans are adenine, guanine, cytosine, and thymine. So these are the bases that are found in humans, and they are all the same. They are all the same, and they are about 4 billion. We have about, 3 billion. We have about 3 billions of these nucleotide bases in humans, and the number are the same, and the bases are identical, but it's just that in some cases, the arrangement of these bases may be differ, or the expressions of the genes from these bases differ from one another, and that is what make us differ from one another. And of course, this is how the nucleotide bases are, and of course, they are in in a cell. We have the nucleus, and then in the nucleus we have DNA. So in the nucleus, we have chromosomes. Then in the chromosomes, we have DNA. And then in the DNA, we have a segment of DNA, which is described as gene. And these genes are made up of nucleotides. And the nucleotides are adenine, cytosine, thymine, and guanine. So then the next thing is genome sequencing. So genome sequencing is the process of determining the complete DNA sequence of an organism or of an organism's genome. So it involves identifying the order of bases. And this order of bases can start from adenine. The order of bases, which is adenine, cytosine, guanine, and thymine, that make up the DNA molecules in each of the chromosomes of an organism's DNA to decode the genetic fingerprint of a human. So the human or the genome sequencing is actually a process of identifying or of determining the complete DNA sequence of an organism's genome. So it involves identifying the order of the bases in the genome, that is in the DNA molecules. So what is a human genome project? A human genome project is a project that was started in 1990 and completed April 2003. It means that it took the scientists 13 years to complete the whole or the genome sequence of human. So this human genome project is an international project. It is an international program that leads to the decoding of the entire human genome. In this human genome project, it actually comprises like 6 male labs in 6 different countries, which include Japan, China, USA, UK, France, China. So these are the countries that actually participate in the human genome project, and it takes the scientists 13 years complete the identifying the order of bases in humans, and that is how they were able to know that there are like about 3 billion base pairs of DNA, and it's from there that we know the order of genes, the number of genes that are found in humans. And of course, this is very interesting project. So the Human Genome Project gave the ability for the first time to read nature's complete genetic blueprint for building a human being. So this Human Genome Project, for the first time, it was able to read the nature of the complete genetic blueprint for human, genetic blueprint for building a human being. So of course, with the knowledge of Human Genome Project, you were able, or scientists are able to know the sequence of bases and then the genetic blueprint for each and every living organisms, and how these genetic blueprint are used in building the complete human being. And then, so what are the applications of this sequencing, or the application of sequencing in general, to know the sequence of bases of a particular living organisms? So number one, it actually helps in disease diagnosis and treatment. in identifying genetic mutation and variations that contribute to disease conditions. So, sequencing of DNA can help in disease diagnosis as well as the treatment of the disease. And then there are one of the major benefit of this Human Genome Project. It's actually when the Complete Human Genome Project was able to was established, nearly about 1,000 diseases are due to single mutation or single gene malfunctioning. So with the application of this sequencing, scientists was able to detect about 1,000 diseases that was actually occur due to single gene malfunctioning. And then it also it fits the code of viruses like in 2014, because when the Complete Human Genome Project was launched, scientists was able to sequence the the DNA of so many organisms, of so many other organisms. Like, for example, in 2014, a group of scientists from MIT and Harvard was able to sequence a samples of Ebola virus from infected African patients to show how the genomic data of viruses could reveal hidden pathways of transmission. And then, what are the applications of Human Genome Project? So, Human Genome Project was able to help scientists in drug development. So, drug development can identify genetic target for drug development and testing, leading to the development of more effective and personalized drugs. Like, for example, generally in drug development process, in drug development process. So therefore, Human Genome Project was able to help scientists in drug development, which actually have in identifying the genetic target for the drug development and testing, which also leads to the development of more effective and personalized drugs. Like, for example, it takes not less than 12 to 15 years to develop drugs, which start from basic laboratory work, in vitro, in vivo, and then clinical trials. So drug development involves basic research, which is in the lab, and then the preclinical states, which is in vitro and in vivo tests, which take 3 to 5 years, and then clinical studies, which include phase 1, phase 2, phase 3. So clinical studies can actually be achieved between 3 to 7 years, and then the drug approval or the approval of drugs, which take 1 to 2 years. So generally, drug development started from basic test tube, started from basic test tube, basic test tube, followed up to the stage of human trials. So Human Genome Project was able to help scientists in drug development. And then in addition, prenatal screening. So with the knowledge of Human Genome Project, it helps scientists to be able to screen babies whether they have any genetic disorders or anomalies. Like, for example, the preclinical testing can be used as a tool for prenatal screening to investigate whether the fetus has genetic disorders or anomalies. So Human Genome Project was able to contribute and help scientists to be able to detect diseases like sickle cell anemia. If you have if a pregnant woman, you can be able to use this prenatal screening or prenatal screening to test whether the baby develop any genetic disease like sickle cell anemia risks of developing hypertension, and so many other diseases. So with the knowledge of human project, scientists was able to do this prenatal screening. And then also in agriculture, Human Genome Project was able to help scientists to identify genes that contribute to desirable traits in plant and animals, allowing for selective breeding of crops and livestock. Like for example, currently there are some GMOs, like some plants, that some genes were able to establish in some microorganisms that have the potential to kill insects. So scientists was able to establish that, or was able to use the knowledge to identify genes like insecticidal proteins from Bacillus thuringiensis. They were able to identify some genes that can kill insects, and they were able to isolate those genes and actually insert them into plant, and then the plant will now develop a trait of killing the insect. So that is GMOs. So scientists was able to do that with the knowledge of Human Genome Project to identify a particular genes that is desirable or that have a desirable trait which can be placed into plant or animals, which actually lead to the selective breeding of crops and livestock. And then in addition, evolutionary biology, it can help to trace the evolutionary history of species and understand the mechanisms underlying evolution. So with the knowledge of Human Genome Project, a lot of DNA or a lot of organisms or different species, the sequencing of the DNA from different organisms, plant, animal, microorganisms was conducted, and then scientists can be able to trace the evolutionary history of some species of organisms and also understand the mechanisms underlying the evolution. Like for example, if you check human being of nowadays and the human being of 1,000 years ago, we are not the same. We are not the same. So with the knowledge of Human Genome Project, scientists can be able to trace the evolutionary history. That is how the living organisms change over time and how different we are, how different we are with those organisms that survived 100 years, 200 years ago. And then forensics. So with the knowledge of genome sequencing, scientists scientists can use this knowledge of Human Genome Project in identifying suspects in criminal investigations and establish paternity in cases of disputed parentage. So scientists was able to use the knowledge of Human Genome Project in identifying suspects for criminal investigation in courts. So what are the relevance of medical biochemistry to other life sciences? So although biochemistry, as we said, is a branch of sciences that is associated with the chemical processes that are taking place in living organisms. So this biochemistry and clinical biochemistry relate with genetics, molecular biology, toxicology, immunology, microbiology, virology, endocrinology, physiology, and pharmacology. It related with all these other areas, like for example, toxicology, study of toxic substances, immunology, molecular biology, genetics. And if you want to if you need more explanation on how medical biochemistry relate with other life sciences, there is a video link. I'm going to drop it on this video. After you watch it, then you can click on it and watch the video to have so that you will have a clear explanation of how the biochemistry or medical biochemistry relate with this other area. Like briefly, we know that biochemistry is associated with physiology because physiology is the study of functions of living organisms. So biomolecules that are found in a cell in each of every living organisms are the one that determine the function of a specific organs or of a specific cells or of a specific tissues. So it's like the biochemistry is the language of physiology, because in biochemistry you are studying biomolecules that lead to the function. So biochemistry is a language of physiology, and then pharmacology is a branch of sciences that is associated with drug development and drug discovery processes. And biochemistry is important in develop or in helping scientists to identify a drug targets as well as identifying the drug's compound. And then genetics, which is the study of heredity and variation. Of course, you cannot understand the knowledge of genetics without understanding the DNA and RNA. And of course, in biochemistry, we fully understand and study DNA and RNA. And that is why biochemists can always give a clear explanation of genetics. And the molecular biology is the study of informational macromolecules, which is DNA, RNA, and proteins. And then toxicology is the study of toxic substances, including drugs and so many other things. So these are how this biochemistry, and of course, biochemistry is directly linked with veterinary medicine and human medicine. So then we are now going to look at the branches of biochemistry. So starting with the toxicology. So toxicology, this field study the adverse effect of toxic or foreign chemical substances on organisms. Like for example, in human, we take drugs. Generally, human take drugs. So drugs itself is a foreign substance and is a toxic. So the study of how these toxic are manipulated in human or in every living organisms is what you call toxicology. So, of course, the area of toxicology is also associated with how the toxic substances are detoxified and metabolized in the body. So, environmental and food toxicology are also fall under this branch of biochemistry. Like, for example, this is how the body of living organisms was able to remove toxic substances. Like, for example, let's say we take acetaminophen, that is paracetamol. So when we take it, it is a foreign substance. It is a toxic substance in our body. So when we take any drugs, the best thing to actually eliminate the drugs after it elicits it is pharmacological activities. And the longer it stays in our body, the more damage it will cause to that particular organs. So generally, drugs are metabolized in a liver, hepatocytes, specifically in the smooth endoplasmic reticulum of the cell. So in the smooth endoplasmic reticulum of the cell is where the drugs are metabolized. So generally in the smooth endoplasmic reticulum of the liver cells is where drugs are metabolized. And of course, after metabolizing, then the drugs will be detoxified, and then the conjugating agent will now bind with the drugs and then eliminate it. So like now, for example, when we take acetaminophen, cytochrome P50, that cytochrome enzymes is the enzymes that will now convert the drugs into less toxic. It will now convert it and it will now convert it to make it more hydrophilic, and then the conjugating agent like sulfur or glucuronic acid will now bind to it and then make it more easily excretable and make it non-toxic, and then it will now be excreted in the urine. So when we take drugs, we have to metabolize it. First, we have two phases of drug metabolism. So the first step of drugs metabolism is to make it hydrophilic. Generally, most of the drugs are lipophilic in nature. Why is it that they are lipophilic in nature? Is because of the membrane of the cell. So the membrane of the cell is actually lipid in nature. So most of the drugs are lipophilic because when we have disease, we want the drugs to easily get into the target organs or the target cells. And these cells are made up of a lipid. So that is why most of the drugs are lipophilic in nature. That is, they can easily be dissolved in the membrane and then transported into the cell. So most of the drugs are lipophilic, that is, they dissolve on the cell membrane and easily get access into the cell. So once they get into the cell, then the first phase is actually to make it hydrophilic in nature using cytochrome enzymes in the first phase. And then the second phase is to make it non-toxic using conjugating agent. And after the body make it non-toxic, then it will now be excreted in the urine. So this is how the toxic substances and the foreign substances like drugs are metabolized in the body. And of course, that is the area of toxicology. And we have so many areas of toxicology. It can be food toxicology, environmental toxicology. And then the next thing is enzymology. What is enzymology? So enzymology is the study of enzymes, their functions, deficiency, and the consequences of such deficiency in diseases. So like, for example, when we said enzymes, because we said enzymes is the study of, enzymology is the study of enzymes. So what are enzymes? Enzymes are biomolecules that speed the rates of biochemical reaction. They speed the rate of biochemical reaction. And how these enzymes work? Enzymes generally have a site called active site. And this active site is a site where the substrate binds. So like, for example, in this case, if you can clearly see from here, you can see that here we have an enzymes. You can see that here we have the active site of the enzymes here. We have it here. And these are the two substrates. So the two substrates now bind to the active site. Then it now gives us enzyme substrate complex. And then the product will be released. So it means that the enzymes now is fully converted the substrate into product. And that is why they have different color. So here we have the free product and then the free enzymes. So enzymology is associated with the study of enzymes, the functions of the enzymes, and the deficiency of the enzymes, because sometimes the deficiency of these enzymes can lead to disease condition, especially like inborn error of metabolism. And then this deficiency, and apart from the deficiency, is also associated with the consequences of the deficiency in the disease and disease processes. So that is this. And then the next thing after the drug, after the metabolism is after the concept of enzymology is to know how these enzymes speed the rate of the reaction, because enzymes generally speed the rate of the reaction, because in some cases you can have a reaction that might take years. You may have some reaction that can take days. You may have some reaction that can take hours. But in the presence of enzymes, the reactions can easily take place within seconds, within minutes. So how does that happen? So in the presence of enzymes, look at it. This is what we call activation energy. So activation energy is the energy barrier that must be overcome for a reaction to proceed. So this is the substrate here. So we have a substrate. We have a substrate here. So this substrate, we have a substrate here. So this substrate will be converted into product. So before this substrate converted into product, then the substrate must actually jump this activation energy before you have the product. So in the absence of enzymes, without the enzymes, the activation energy is high. So it means that the substrate needs sufficient amount of energy before it converted into product. But what enzymes does, actually, the main things that enzyme does is actually to lower the activation energy. So you see in this case, the activation energy is lower here. So that is why the substrate can easily be converted into product. So it means that Enzymes speed the rate of the reaction by lowering activation energy. So ladies and gentlemen, if this is the first time you are coming, I encourage you to try to subscribe to this YouTube channel and register with my website. It will really help. So then the next thing apart from the concept of enzymology is another branch. So we have another branch of this enzymology. So we have molecular biology and biotechnology. So molecular biology, this branch of biochemistry, or this branch is evolved directly from nucleic acids biochemistry, and it involves manipulation of DNA to improve drug research and solve health problems. It has wide application in other fields of science, which include cancer research. So biotechnology and molecular biology is actually a branch of chemistry. There are two main branches of biochemistry that are associated with the manipulation of the DNA. And this manipulation of the DNA can help to improve drug research, also solve problems that are associated with the health problems. So now let's look at one of the let's now look at one of the branch, which is molecular biology. So molecular biology is an interdisciplinary approach which help in investigating the molecular mechanisms and dynamics of living organisms. So when we said molecular biology is a branch of biochemistry that is associated with informational macromolecules. That is associated with informational macromolecules. These informational macromolecules includes DNA, RNA, and proteins. So in molecular biology, we are dealing with these three macromolecules: DNA, RNA, and proteins, and the mechanisms as well as the dynamics in living organisms. Like for example, now let's look at, you see, generally in living organisms we have DNA, and this DNA is converted into RNA through transcription, and then from the transcription it will now be converted into proteins through translation, and then the proteins undergo folding. Then you will now have a functional protein, and this functional protein is what will determine the functions of the body of a living organisms. Like for example, red blood cells is responsible for the transport of oxygen due to the presence of hemoglobin. So proteins is what determine the structure and the functions of a cell. Like for example, cells have different shape, cells have different functions, and all these differences is due to the different proteins that are produced in different cells. So the study of DNA, RNA, proteins in a living organisms is what we call molecular biology, as well as the mechanisms that lead to the change of these molecules from one form to another. And then the next one is the biotechnology. So biotechnology is also a branch of science that combines biology and technology to create a product and services using the living organisms or their fats. So generally in biotechnology, we produce or we create a product. It can be drugs, it can be vaccine, it can be industrial products, like for example, insulin produced through using this biotechnology process, oxytocin that we are injecting patient, the insulin that we are infecting diabetic patient, they are all created through the knowledge of this biotechnology. So in biotechnology, you are using the whole living organisms or the fats of the cells, like for example, or the part of the living organism. It can be cells, it can be a protein, it can be tissue, it can be the whole organism. So if you are using cell, or if you are using the whole organisms, or if you are using tissue to produce a product that will be of beneficial and importance to humans, that is what we describe as biotechnology. And we have different types of biotechnology. We have red biotechnology. So a red biotechnology is a type of biotechnology that involve, whether it's associated with medicine or that is associated with medical and pharmaceutical industry. And then we have white or gray biotechnologies, another types of biotechnology that is associated with industry. We have green biotechnology, which is associated with agriculture. We have gold biotechnology, which is associated with bioinformatics and data. And then we have blue biotechnology, which is associated with marine and aquatic environment. We also have yellow biotechnology, which is associated with food production. We also have violet uh, biotechnology, which is associated with governance of ethical considerations. And then we have dark biotechnology, which is a warfare. So you see, these are the different areas or different types of biotechnology. Like, for example, in green biotechnology, is a biotechnology that is associated with the production of agricultural products. Red biotechnology is a type of biotechnology that is associated with the production of drugs, vaccine, and other chemical substances that can use in disease treatment. While white or gray biotechnology is a biotechnology that is associated with the production of industrial products. So these are actually, and then warfare, which is a dark biotechnology, is a biotechnology of bioterrorism, where scientists can create a virus or a bacteria or any living organisms which can be used to actually cleared or maybe virus or any things or any organisms that can be used in war. Yes. And then the lipid and carbohydrate biochemistry. So these are the fields that study the chemical basis of metabolic disorders such as diabetes, obesity, and cardiovascular disease. So generally, when we take too much lipid and when there is high carbohydrate content in our body, it associated with disease such as diabetes, obesity, and cardiovascular disease. Like, for example, if our blood glucose level is high and insulin are not actually properly produced or cannot properly utilized by the body, then the blood glucose level will go very high and that will lead to diabetes. And then obesity is when when we have the high fat content in our body, and that will lead to obesity. And of course, in biochemistry or in nutrition, we can know someone is obese based on or using BMI, that is body mass index. You can measure the weight and divide it by the square of the height of the particular individual, and from the value you get, it will tell you whether the person is obese or not non-obese. And then cardiovascular disease is a disease that is also associated with high content of lipids, and also, or that, like, for example, when the cholesterol level is very high, which is also an example of lipids, then it leads to a condition called atherosclerosis, which lead to cardiovascular conditions or cardiovascular disease. So these are the examples of the lipids. A lipid can be cholesterol, it can be triglyceride, it can be phospholipid. These are some of the examples of lipid. And then for the carbohydrate, we have different types of carbohydrate. We have simple carbohydrate, we have complex carbohydrate. Simple carbohydrate, it can be sugars like fructose, sucrose, glucose are the example of simple sugars. And then we have complex sugars, which can be starch and fiber. So these are some of the examples of carbohydrate. And as we continue with the lectures, we are going to look at all these classes of lipid and carbohydrate with time. And then the next branch of biochemistry is natural product biochemistry. So this is a new area of research in biochemistry. It evolved as a result of interest of scientists across the world in searching of new drugs from plants, microorganisms, or from animals. So generally, scientists are trying to use a compound from natural products or from plants, animals, microorganisms to identify or to search for new drugs which can be used in the treatment of different diseases. Like, for example, quinine and artesunate are antimalarial drugs and were isolated from plants. So the natural product biochemistry is a branch of biochemistry that is trying to identify drug substances from plant, and that is natural product biochemistry. Like for example, this is a natural product from microorganisms. We have antibiotics, penicillin and streptomyces. They are antibiotics that are actually obtained from microorganisms. There is also anti-tumor drugs, and remycin is an example of a natural product from microorganisms. That's a chemical compound that was able to be isolated and identified from microorganisms and is used in treatment of cancer. And then other metabolisms like lovastatin is also a drug that was actually identified and is used in the disease treatment. So ladies and gentlemen, apart from these branches of chemistry, biochemistry, we have so many other branches, and I'm going to actually send you another link which will help you to see other branches of biochemistry like structural biology, cellular biochemistry. There are so many of them, and I'm going to share the link for you to see the branches, to see other branches of biochemistry. But these branches or this lecture specifically come from your lecture materials. So thank you very much for attending this lecture, and please subscribe and join our YouTube channel. And if you are interested, you can visit our website rrfasch.com.ng and register with our online courses. Thank you very much till we meet in the next lecture.